Let's talk about acquired immunity in some greater detail. For review, our immune system is broken up into innate immunity and acquired. Acquired is also known as adaptive immunity. We could also describe these being non-specific and specific. That is to say, the innate immunity is non-specific. It fights pathogens, it fights toxins, anything foreign entering the body, but not specifying what that foreign entity or agent is. Whereas acquired immunity is focusing directly on a particular pathogen, whether it's chickenpox, smallpox, rabies, COVID, influenza, any number of pathogens. Now, it is totally incorrect to separate innate and acquired immunity, but that's what we tend to do when we're teaching a subject like physiology. We actually delineate between 11 body systems, but all our body systems don't work independently. That is to say, our cardiovascular system, the aim of the cardiovascular system is to distribute oxygen-rich blood and nutrients to the tissues. It utilizes the heart, the blood vessels, and the blood, but it's not independent of the respiratory system. That is to say, the cardiovascular system would be totally meaningless without the respiratory system bringing in oxygen. And it would also be silly not to include the importance of the renal system, because the renal system filters our blood. My point with this, it is challenging when we teach physiology or the immune system in a reductionist manner. That is to say, breaking things down into separate parts, because the body doesn't really work in separate parts. But sometimes it's easier to explain things, and that's why we do it, and I'm going to continue doing it this way. But my point here is, please understand that innate immunity and acquired immunity are both trying to achieve the same means, making our body safe so we can maintain homeostasis. And there is some overlap. So acquired immunity, once again, is adaptive immunity, and it's specific. And acquired immunity is generally or broadly broken down into humoral immunity and cell-mediated immunity. And we've talked about white blood cells. We haven't talked about lymphocytes. Lymphocytes are white blood cells, but they are specific to our acquired or adaptive immunity, or our specific immunity. So B lymphocytes, otherwise known as B cells, are specific to our humoral immunity. Humoral or humor is referring to our blood plasma. Back in the 15th, 16th century, when people were first studying human biology, they referred to any body fluids as humors. Hence the term humoral immunity, which we still retain when we're talking about blood. And we really only retain it when we're talking about the immune system, specifically antibodies. Antibodies are plasma proteins that flow through the blood and play an integral part to our immune system. We'll talk about that momentarily. Antibodies are released from B cells or B lymphocytes. The other portion or other component of the acquired immunity is cell-mediated immunity, and that's the role of T lymphocytes or T cells. So we'll talk about all of these. The basis for acquired immunity is antigens, and these are cell surface proteins or very large polysaccharides on pathogens that our body is cueing off. We are recognizing these specific antigens. Now remember, when we were talking about innate immunity, things like neutrophils and macrophages could identify or do identify things as foreign based on pathogen-associated molecular patterns. Just common themes they find on or any infectious agent. But antigens allow our immune system to zero in on specific pathogens. And antigen really is short for antibody generator. So it's producing antibodies based on this surface molecule around the pathogen. So these are representing antigens here. Antigens are merely identification tags denoting something foreign. And it would be wrong for me to suggest we never use the term antigen for anything endogenous. That is to say, anything from our own body. Because when we look at the ABO blood group or describe the ABO blood group, we refer to the cell surface markers as antigens. So antigens are not specific to foreign agents, but they almost are. And another component of acquired immunity is memory, and this is a huge component of acquired immunity. That is to say, if we get infected with some sort of pathogen, our body is going to remember that pathogen. And subsequent infections is going to lead to a much more rapid response, a much more robust response, and a much longer duration of that response, months rather than maybe days or weeks. So to be clear, the acquired immunity is so valuable and so effective that we are able to withstand very, very, very high infectious doses of pathogens into our body, upwards of 100,000 times more than the lethal level. My point being is that our immune system, our acquired immunity, is so effective that it can withstand very high doses of infections. So let's take a look at antibodies. Antibodies, once again, are plasma proteins, and they are also known as immunoglobulins. The main type of antibody specific to our acquired immunity are what we refer to as IgGs. There are also IgMs, which are really the first antibodies produced during an infection, but ultimately they're going to turn into IgGs. Don't worry about the dynamics of that. And then IgEs are parts of allergic responses. One thing I do want to back up and say about acquired immunity is that, once again, we do have memory. So an example of that, when I was in 3rd grade, I got chickenpox. I never got chickenpox again. Certainly I was exposed to it again because there were other kids in my class getting chickenpox in the 3rd grade. There were probably some getting it in the 4th grade, maybe 5th grade, 6th grade. Who knows when it ended? But I was still exposed to chickenpox, but I never got sick again because I had memory, and our memory cells, whether they're memory B cells or memory T cells, act much, much, much more quickly, dare I say immediately, to eradicate the body of that infection in the case of chickenpox or varicella. Varicella and chickenpox are the same thing. So back to antibodies. Antibodies are released by a type of B cell known as a plasma cell. We'll get into this in a few seconds. Antibodies bind to antigens. So here's an antibody. There's a few different portions to an antibody. The vertical portions here are what are referred to as the constant portion. They tend to be constant amongst all antibodies. But the Y part of it, the top part, is the variable portion, and that's what changes or is modified to specific pathogens. So we see these aren't really pathogens, these are the antigenic components of the pathogen. They could be complete antigens or they could be fragments to it. But what we see here is now this antibody, which can always bind, at least in this case, to 2 antigens. This is what's known as the antigen-antibody complex, and that's going to elicit some sort of immune reaction within our body. So what does the antigen-antibody complex do? It causes agglutination, which is clumping of these pathogens. So we can see antibodies here binding to a number of different pathogens. That clumping is going to allow or facilitate a more efficient immune reaction. That is to say, white blood cells will come to the area and attack this clumping of pathogens here. Precipitation is very similar. It is where the pathogen, the antigen-antibody complex, is no longer soluble in fluid. As we see right here, it's very similar to agglutination, but allows for our white blood cells to access the antigen-antibody complex. Lysis is rupturing of cells, or in this case of the pathogens. So to a certain degree, antibodies can facilitate lysis. Neutralization, they tend to block the sites on pathogens that release toxins. But arguably, the single most important aspect of the antigen-antibody complex is turning on the complement system. The complement system is a system that kind of balances the midway point between our innate immunity and our adaptive immunity. So the complement system is composed of approximately 20 plasma proteins. So we have a whole host of plasma proteins floating through our blood. We've talked about things like fibrinogen and fibrin. Fibrin is what causes blood to clot when there's some sort of laceration. But we've also talked about we don't want fibrin activated all the time. That is to say, if there's no hemorrhage, there's no laceration, there's no need to activate fibrin. We don't want our blood clotting when there's no problem. So we don't want fibrin continually flowing through our blood. That's why we have the inactive form of it, which is known as fibrinogen, which will then get converted to fibrin. And it's similar in the complement system. We have 20 inactive plasma proteins, but under the right conditions, they will become activated and stimulate some downstream events. So what happens is the constant region, once again, that's this vertical part of the antibody, antigen-antibody complex, that binds to the first protein in the series, which is known as the C1 protein. And the C1 protein is going to initiate a whole cascade of events. Now, to be clear, I am simplifying this dramatically. And one thing you will notice here is there is overlap and repetition in a lot of these processes, which is important because if one aspect of our immune system is not functioning well or is not getting the job done, hopefully another aspect will. So it is also going to facilitate or cause agglutination. It's going to release cytokines. And when I say it, I'm saying the complement system, because there's a whole host of proteins here, 20 some odd proteins that have different jobs. But one protein is going to stimulate another protein, which is going to stimulate another one. And it actually may not be the proteins, it may be cytokines released from these proteins. Cytokines are chemical mediators or modulators facilitating some sort of metabolic action or activation of cells. Chemokines are a type of cytokine that attracts other white blood cells to the area of infection. And chemokines will facilitate chemotaxis, which is, as I've described previously, throwing those breadcrumbs out to attract other white blood cells to the area they need to go to. Additionally, this whole complement system will activate basophils and mast cells. Once again, basophils and mast cells are very similar. The only difference is mast cells reside within the tissues, whereas basophils are free-floating in the blood plasma. They're going to release histamines that cause vasodilation and increase capillary permeability, and that's going to lead to inflammation. This complement system is also going to activate a lot of phagocytic cells, macrophages, dendritic cells, neutrophils, and there's also a component known as the lytic system that causes cells to rupture, essentially explode. Let's take a closer look at B cells and how all of this plays out. B lymphocytes, once again, we can refer to these as B cells, and I will refer to them as B cells. B lymphocytes are synthesized in our bone marrow. You can remember that B for bone. And B cells or B lymphocytes function as antigen presenting cells. So what we see right here is a B cell functioning as an antigen presenting cell. What happens is the B cell has the antigens bound to its receptors. And to be clear, the receptors on B cells, the receptors for antigens, are antibodies. I believe most people think of antibodies as free floating in our blood, which is true. But the receptors on B cells are those antibodies. So if we have antibodies for varicella or chickenpox, those are free-floating antibodies in our blood plasma. But the B cells specific to chickenpox have antibodies on their cell surface. They are cell surface receptors. You could call them B cell receptors. But here's a B cell, and it has these antigens bound to its receptor, which is the antibody. And that is then going to present this to our T cells, which are going to really do the work to destroy and kill the pathogen. We previously mentioned that B cells do play a part and antibodies do play a part in neutralizing and potentially destroying pathogens, but really the workhorse for destruction are the T cells, specifically cytotoxic T cells. How do these B cells get activated? This is amazing and complicated, but what happens in embryonic and fetal development, we start getting our lymphocytes being developed, and they develop their surface receptors, antibodies, B cell receptors, and the same thing with T cells, T cell receptors. And they're going to produce, that is to say, the immune system is going to produce thousands and thousands, maybe even millions of cells with different receptors on them. Each one of those receptors is going to be specific to a pathogen, specific to smallpox, specific to varicella, specific to rabies, influenza, COVID, what have you. So there's going to be a whole host of these, and we'll talk about in a moment how these become immunocompetent. But what happens when the specific antigen binds to its corresponding receptor, in this case on the B cell becomes activated, and that B cell is going to divide. One thing I want to point out, B cells and T cells have a limited lifespan. They're eventually going to die off. And when I say a limited lifespan, I'm talking about specifically the ones that are carrying out the work, the ones we refer to as effector cells. So the activated B cell is going to divide. One thing it's going to produce are the effector B cells. Those are the ones that are going to carry out the work that B cells do, and that is release antibodies into the blood plasma. The effector B cells are called plasma cells. So plasma cells are nothing more than the cells that carry out the work of B cells. Plasma cells are B cells. They are specifically the effector B cells. Effect merely means facilitating or carrying out some sort of process downstream, or doing some work or affecting some pathogen downstream. But when the B cell divides, because as we said, these effector cells are eventually going to die out, when the B cells divide, they're also going to create memory cells. And this is super important for adaptive immunity for subsequent exposure. Once again, subsequent exposure, our immune system is more robust, much quicker, and lasts for a longer duration. And that would not be possible unless we had these memory cells. Lymphocytes are similar, but they are synthesized in the thymus gland. The thymus gland sits just above the heart in the mediastinum. This is a bit high, so maybe right here. Right in front of the great vessels, the aorta and pulmonary artery, immediately anterior to them. With the case with B cells as well, while B cells are synthesized in the bone marrow and T cells are synthesized in the thymus gland, they both migrate to lymphatic tissue, which are lymph nodes, the spleen, and mucosa-associated lymphatic tissue, and some other regions within the body that have high congregations of white blood cells. The receptors on T cells are very similar to receptors on B cells. We call these T cell receptors, and I realize that's not an exciting name for them, not as exciting as an antibody, but they are antigen specific. That is to say, only one specific antigen is going to bind to these T cell receptors. If there's a T cell specifically to varicella, smallpox is not going to bind to it, COVID's not going to bind to it, influenza is not going to bind to it. It's just going to be varicella that binds to it. There are 3 types of T cells. 2 of them are regulatory cells, the helper T cell and the repressor T cell. And the third one is the cytotoxic T cell, which is the one that is really responsible for eradicating and killing infected cells, cancer cells, and virus-infected cells. They're really destroying our infected cells. They destroy, they eliminate, they poke holes via secretions known as perforins. They poke holes or put perforations within the pathogen, which causes solutes to move into the pathogen. Water follows via osmosis and causes that cell to lyse or explode. T cells are going to release toxins as well, aimed at neutralizing or killing infected cells. And cancerous cells are normal in all of us, but they tend to be regulated by a number of aspects of our immune system, one of which are cytotoxic T cells which will kill or destroy cancerous cells that are dividing way too much. Helper T cells are super important. They're going to stimulate not only other T cells, but B cells as well. Individuals that are afflicted with HIV/AIDS, their biggest issue is lack of helper T cells. And without helper T cells, the acquired immunity is not up to snuff. It really can't do its job without the helper T cells. Repressor T cells, sometimes referred to as suppressor T cells, make certain that our cytotoxic T cells and helper T cells are not overactive. So they're making sure that there's some sort of stability within our acquired immunity. And the T cells, like B cells, are going to divide into effector cells and memory cells. So upon infection, that antigen is bound to the receptor here. This is a T cell receptor. It's going to activate the cell, and the first thing that cell is going to do is divide, make a bunch of effector cells and make a bunch of memory cells. The memory cells are saved for a later date, just like the memory B cells, and then the effector T cells are going to go out and carry out their work, whether they're helper T cells, repressor T cells, or the cytotoxic T cells. Very early in development, we get millions of B cells and T cells with different receptors on for specific pathogens. But one of the single most important processes that plays out is what's known as immunocompetency. And that means this is teaching our B cells and T cells not to recognize our own cells as foreign. And basically what happens here is that if one of our naive lymphocytes, if it binds to one of our own cells, this would be one of our self cells. It doesn't matter what type of cell it is. It could be a liver cell, a bone cell, a skin cell. If one of these naive lymphocytes binds to one of our own cells, that cell is destroyed, and it's no longer going to divide and create progeny. That is to say, we don't want any cells binding to our own cells. We want them to be immunocompetent and know the difference between self and non-self. Herd immunity is a great way to understand the acquired or adaptive component of our immune system. So if we look at this right here, in light blue, which we see right here, is an immunocompromised individual. Immunocompromised individuals are much more susceptible to infectious disease than other individuals in the population. In red, these are individuals that are becoming infected. In dark blue, these are just people that are not immunocompromised and they haven't initially been infected. So what happens in a population of individuals that have never been exposed to a specific pathogen before is that infectious disease will spread throughout that population. Now, certainly I've got a couple individuals in here that did not get infected. In red is infected, but it tends to spread like wildfire if no one in the population has developed or acquired any immunity to that pathogen. Some of them are going to get infected, some of them may get sick, but the immunocompromised are going to be worse off and potentially die. Now, certainly some of these red ones could die as well, depending on what the infectious disease is. But if within a population there are individuals that have acquired the immunity because they've been exposed to that pathogen before, it is going to protect a large component of the population, but most importantly, it is going to protect those immunocompromised individuals. So in this population right here, let's suggest these two red individuals who are infected were never exposed to this pathogen, whatever it is. So because they were never exposed to it, they did not develop the acquired immunity. That cannot be spread to the immunocompromised individuals. And these are people who are too young, who are super young and maybe don't have a fully developed immune system, people who are on the older end of the spectrum and their immune system has become weakened. It could be individuals with certain comorbidities, certain diseases that may be impacted by an infection. But what happens here with herd immunity is if the majority of the population has accrued that adaptive immunity to this pathogen, it's going to protect, it's going to actually put a barrier around this one individual. Immunocompromised individuals generally do not want to get the infection, whether it's received naturally or whether it's received via a vaccine. So vaccines are a safe way to expose individuals to an infectious disease so individuals can develop acquired immunity from the vaccine. For some immunocompromised individuals, vaccines may actually be too much for them. But if the majority of the population has the acquired immunity, we now have what's known as herd immunity. That is, the great majority of people are protecting those who are most susceptible. So this is a form of altruism where individuals in the population are actually protecting those that cannot get the infection. Vaccines work. So if we look at this table right here, this just shows how successful vaccines have been in the United States to certain infectious diseases. And if we look at the top here with smallpox, smallpox has had 100% reduction in its incident in the United States. Now, to be clear, this table is from 2016. Things may have changed since 2016, but I imagine they're still pretty much the same. We can look at measles. There's been almost 100% of reduction, 99.98. Mumps, 98.9%. And rubella, and I'm skipping down to rubella, that's 100%, because a lot of us get the MMR vaccine, measles, mumps, and rubella. Tetanus, just below 100%. So this shows vaccines work. Now, there was a study in a British medical journal back in 1998 where 13 doctors claimed that ingredients within vaccines were causing autism. And that ingredient was thimerosal, which is a preservative for vaccines. Since that time, that's roughly 2+ decades, all the medical research has not shown any impact of thimerosal in vaccines or anything in vaccines that cause autism. 10 of those original 13 authors have retracted the statement saying that thimerosal in vaccines cause autism. One of the main authors on that paper actually lost their medical license because of that paper, suggesting that vaccines cause autism, or in general that vaccines are dangerous. To be clear, vaccines can cause problems in immunocompromised people, and probably in very rare cases may cause problems in healthy individuals. But by and large, vaccines are very, very safe and very, very effective. My aim is not to convince people to get vaccines. I am just trying to share what the scientific research has shown. Unfortunately, there's a lot of hurdles with vaccines because of political ideologies and social beliefs and religious beliefs, and everyone's entitled to have those. But as we see with herd immunity, a lot can be achieved in keeping our global population safe with vaccines.